All right. Hi, everyone. I'm Lynn Pice. I'm an analyst at the Broad Institute and also the product owner for seqr, which is our genomic analysis platform that we developed at the Broad. And it was mainly developed to support large-scale analysis of rare disease data. But since we realized how impactful it has been, and it has been made available more widely to other researchers and clinicians via AnVIL. To orient you to the state of rare disease diagnosis, there's over 300 million people that are worldwide affected with rare disease, and of those, about 30 million within the United States itself. Typically, exome or genome sequencing is the first line of testing, but depending on the condition, the majority of individuals remain undiagnosed. A lot of this data or individuals then sign up to research programs, hoping to find an answer, and what this leads to a large amounts of data being available for analysis, and really the need to efficiently manage, analyze, and store the data. As you're aware, with traditional approaches, most researchers typically have a copy. Each researcher has a copy of the data, and then that needs to be duplicated among researchers, and also need to have individual copies of the tools, and this can be really expensive. And that's one of the reasons the AnVIL app space was created to democratize access to these research tools. For researchers using commercially available platforms, there is additional consideration. For example, they could be expensive. And if they're bought over by an additional company, that leaves open questions to what happens to the research data. And when it really comes to rare disease analysis, one of the critical pieces is being able to collaborate with other rare disease researchers, being easily able to query and share data. And that's the reason we develop, reasons behind developing seqr. As I mentioned, seqr is available on the AnVIL platform and has been there for over 5 years now. So it makes available all these powerful filtering tools on the cloud. And it's been used by over 1,000 researchers, not just within the US, but internationally as well, which includes just clinicians and researchers, as well as medical centers. Here's just an overview of the main features of seqr, which really it promotes collaboration. So if there's any data within seqr itself, the case can be shared among multiple researchers within a team so they can all work together to analyze a specific case. We have over 70,000 samples in seqr as of now, and it's incredible that how the pace at which it's grown, because roughly half of this data is internal Broad data, and the rest is other AnVIL users who've deposited their data and are actively analyzing these cases. With the large amounts of data, it has generated 500 million unique variants, which has been really useful in assessing variants when it comes to interpretation, thinking about whether this variant could be relevant to your case or not. And that's just the power of having large data sets. These stats here, it just talks about data sharing because seqr is connected to the Matchmaker exchange as well as VLM. And that's really helped us had pretty good diagnostic rate. And most of these numbers over here are just from our internal data because we've been able to track it. It's just it's not as easy to track external stats. All right, so there's two main ways that you can interact with seqr. I'll be focusing on seqr and AnVIL for today's demonstration. So seqr and AnVIL is managed by the Broad. There's no cost to using seqr itself. So when you're getting started, you would typically have your data, your VCFs within an AnVIL workspace. So there might be minor cloud storage costs at that stage. And then if there's any compute that you use to join call your VCFs. But after it's transferred to seqr itself, there is no cost to you. You can also have a local deployment. And this is here you would be responsible for all costs and for deployment as well as all the maintenance associated with it. And this is typically recommended only for institutions where they cannot share their data on the cloud, on a cloud platform. All right, so if you were thinking of getting started with seqr, you have a genomic exome or genome data, VCFs. We require a join call VCF. So we do make available WDLs, or if you have your own join calling platform, you'd require this VCF as well as a pedigree and stored in an AnVIL workspace. So I know most of you are familiar with AnVIL workspaces. You just upload it here. And then you would then select Analyze in seqr. This kicks off a fully automated data loading pipeline, which includes quality checks. So if there's any, and that's the reason we do this early on. So if there's any issues with format or missing fields, you get an automatic notification letting you know what's required. And typically, this entire process, so if you have about 50 to 100 exome cases, it's relatively quick and it typically completes under a day, but it really depends on the queue. But we let people know that your data will be available within a week. I won't go into details of the data loading process. We have a dedicated video for that on YouTube that you can always access, as well as on our FAQ page, all the details required to load your data to seqr. Once your data is in seqr, if you want to save storage costs, you can delete this VCF, but that's really up to you. If you would like to minimize your costs, you could then go to Analyze in seqr, which will take you to the seqr project. But you can also directly go to seqr, so I will demonstrate that here. Are you seeing my seqr page? Perfect. Okay. So yes, you can go directly to seqr.broadinstitute.org. And this is the landing page where you see all the projects that you have access to. There's additional features here to see summary data stats, feature updates where we highlight new features, and I'll talk about that later today. We have FAQ, and we also recommend reviewing the privacy policy and our terms of use. And at any time, if you have any issues with using seqr, you can submit a ticket or even a feature request that you think would benefit other researchers. We'd love to hear about it. All right, so let's take a look at one of the projects here. Yes, and also to get started with seqr, all you do is create an AnVIL account again, which most of you, I'm sure, have already. And once you do, you just log into seqr and you can access the demo project that's always available to people if they'd like to get a sense of what it's like to analyze a case yourself. But I'll demonstrate some of the features here. So for today, I'll open up our Gregor training project. And this is what the project page looks like. Since we handle and a lot of researchers work with large cohorts and you have frequent reporting, especially to the NIH, we have pretty strong project management features. So on here, top here, you can see all the families that have been loaded. You get an overview of the analysis status of all of your cases. You can easily navigate to the AnVIL workspace. Any notifications, so such as when data has been loaded to the project, can be viewed here. And then we use what's called a tagging system. So if there's a variant of interest, we put a label on it and what we call a tag. So you can have different tags for variants that have been pathogenic, that have been returned to the family, or the validated or pending validation. So you can use this to manage different cases and projects at different stages of the analysis cycle. If you scroll down, you get to see all the families here. So within this training project, there are 14 families. And you can get an overview. So here's the analysis status. This one has been marked as no clear candidate, which means it's still undiagnosed. So maybe you can go into your pool of cases for reanalysis at a later date. You can also mark cases that you've analyzed, and this can track who's analyzed the case and at what time. It's all time stamped. The type of data and overview of the phenotype, and if there are variants saved in your project, you can get a sense of that here. Let me open up one specific case and show you what an analysis workflow is typically like. So I'll open up family 13. And this is what we refer to as the specific family page because you have all details relevant to this particular family or this case. So on the left-hand side here, you have the pedigree. You have more project management features, such as if a person has analyzed a case, you can just check that off. So I'm just going to do that again for today. Again, it's all time-stamped. If you're working with multiple people in the team, you can assign an analyst. Lots of fields to add your case notes, any analysis notes. Lots of additional fields here, and the phenotype is captured in HBR terms. So that's all listed over here. We have the type of data that's loaded for each individual. So these two here above was the proband, and here we have for the parents additional fields if you'd like to enter that and capture that information. So typically when an analyst is getting started on a case, they would view all the relevant information to get a better sense of like what's going on with this case and how they would approach their analysis. Once you're ready to dive into the data, you can use the variance search. And that brings up this page here. So many of you may be familiar, worked with exomes and genomes. In a typical VCF in a genome file, you'd have over 100,000 variants. A lot of them are maybe rare benign variants, noise, artifacts. So different ways of filtering down that data is by how frequent it is in population databases or the quality score. So we have all these different parameters over here, which are highly customizable. But of course, it can be tricky when you're doing things, you want to do things relatively quickly or you're relatively new to it. So what we've done is we've standardized searches based on several internal tests. We've broken them down into the inheritance based, and we have like restrictive searches as well as permissive searches. So we typically recommend starting like a restrictive search. So for here, for example, I'm just starting with a de novo slash dominant, and essentially is if the parents are unaffected, it automatically defaults to de novo. You can customize all of these fields. So for inheritance, for example, if you have a suspicion that maybe the mom is potentially affected, you can change it to potentially affected. I'm just going to move that back to unaffected for now. Pathogenicity, so if the variant's present in ClinVar or not, marked as path or likely path. Annotations, for example, if you're interested in loss of function variants only or a specific type of variant, you can select what type of variants you want returned. There's a ton of intronic variants, so that's typically not selected. However, we have a Splice AI cutoff score, so any variant like deep intronic variants that might be predicted to affect splicing, that would be returned in your search. In silico filters, we don't typically use these routinely as a cutoff score, but you can add them in if you'd like. Frequency, we rely on Nomad as well as the large internal seqr database, and really having 70,000 samples in there in seqr has been really useful to assess variants and if they're present in affected and unaffected individual. As for the location field, if you have a hypothesis as to what the gene could be or genes could be, you can specify your genes over here. However, for this particular case, I just don't know. So what I could do is I could come up with a gene list myself. I could look at all genes. But to simplify your search, you can create your own gene list and pull it up from here, but we also connect you to panel app. So for example, I know this person has some level of intellectual disability, so I'll selecting a panel app gene list. A panel app is this particular is maintained by Panel App Australia, where they curate genes and the disease evidence. This auto-populates, so you always have an up-to-date gene list. I can further narrow it down by only genes associated with a dominant mode of inheritance and rule out genes that have very limited or disputed evidence. Furthermore, restrictive searches typically focus on high-quality variants, but if for some reason I did want to change it, I could always change these parameters. Finally, if I wanted, I could just maybe rule out benign, likely benign variants, and then run my search. I made some customizations here, but you can run the search as is if needed. So that just took under a few seconds, and it went down from like over 100,000 variants and just gave you one variant. Let me orient you to what you're seeing here. So there's one variant. If there were several results, you can filter it by, I've chosen Inomim because I want to see genes that are associated with disease first, but you can choose whatever you'd like. So here there's a pathogenic variant, and here in ClinVar, and it has this red background just to capture attention that this variant has been classified as pathogenic. It's in the gene RNU42. We have links to several other resources and it's all variants and data here is highly annotated to help you quickly make a decision as to whether this variant requires further assessment or is not relevant to your case. We have information from Omen stating that this gene is associated with Renault syndrome and associated with autosomal dominant mode of inheritance. This column here has lots of features on the variant itself and transcript information. So there's multiple transcripts it would show here. So this is a non-coding exon transcript. We have links out to the UCSC browser. Additionally, if you want to look up the specific variant in other databases or do additional literature searches. Down here, we have the genotype. So here you can see in the affected individual is de novo, absent in the parents. If you hover over, you can get additional stats on the quality. If for some reason you weren't sure about the quality and you wanted to quickly assess it, you could also have your CRAM stored in the AnVIL workspace and load it to a particular case. So you can pull them up really quickly when you're doing your assessment. So here you can see it's a frameshift variant in the proband. So here's the proband and below are the family members, and it's absent. The reads are pretty clean. I'm pretty confident that this is a real variant. Typically, you'd have in silico scores here, and for Splice AI, we use a pre-computed file. So for this particular variant, there wasn't a pre-computed score, but you can run it up on the fly, which just links to one of our other pages, and you can set your thresholds and get a Splice AI score. And finally, on the right-hand side is frequency in population databases. So as here you can see it's absent in Nomad, but there are 18 individuals within seqr. And what's really powerful is what we show you is how many individuals have the variant and are affected. So that's great to assess, know that this variant is present in affected individuals. If you want to get more information who these affected individuals are, learn more about that, you can use our love to new feature that's been proven incredibly useful is the variant lookup. So you can look up all the variants in seqr. And this is independent of what not just your project, your internal data, but all data within seqr. Now, you can use this variant lookup feature. You don't have to have your data in seqr, and just in case you were interested in another variant, you can always go to the summary data page and variant lookup, put in the variant details, and you'll get this level of annotations. What the variant lookup does is not only looks up internal variants, all those 18, but it also looks up additional databases that are connected to the variant level matching effort. But this is currently absent, or at least VLM does not support indels for now, so we just don't have that information. But if you scroll down, you get into an insight into who those 18 individuals are. So here's the case family 13. That's our case that we were looking at. But you can see here it's the variant's present in several affected individuals here and is de novo in all of these cases. So we just make available high-level de-identified information when it pertains to other cases. You can hover over and you can get a general sense of the HPR terms, which are just high-level information that we make available. You're interested, you would like to reach out to this person, you can just use the contact feature, which auto-generates this email, all the details about the variant, including your name, and you can edit it and send out this email, which goes out to the researcher. So that's about variant matching. And you can, so this is here in Family 13, we did a case-specific analysis. But anytime, if you wanted to just look through all of your data, what you can do is just go to the search. It opens up this page. So for example, you were attending a conference or reading literature and you came across this new gene and you want to query all your cases at once. So I can just choose the entire project. So I'm just selecting the entire training project, all 14 cases. Maybe I want to query all external projects at the same time and only focus on my unsolved families. I can do that. I can run the same search, type in the new gene that I found and query all of the cases simultaneously. Same thing, you can run a similar search. For example, you have just loaded 200 samples to seqr. You're running maybe the cohort is limb-girdle muscular dystrophy, so you can put in all genes associated with limb-girdle muscular dystrophy and quickly highlight just the variants in those specific genes that might be relevant to your cohort. Once you've solved those cases, and then maybe you can go case by case into the other cases that require detailed analysis. So we make available a large amount of features that can really help the rare disease researchers to quickly and efficiently analyze cases family-based, but also cohort-wide. All right, let me just transition back to my slides for now. Yes, so I talked a little bit about variant level matching, but I just wanted to highlight that if you have data sets that you think, what you would want other people to query and be part of this, you can definitely reach out to VLM. It's part of the GA4GH. The other thing that seqr provides in terms of data sharing is Matchmaker exchange. So the VLM earlier was the variant level matching, but this is at the gene level. So there's several nodes here, as you can see, including the Broad Institute. That's part of these nodes. So if there was a gene of interest, here's the seqr page that it's integrated into Matchmaker Exchange. It pulls up all the relevant information about a variant. So for example, here, BIC1, I want to learn more about this gene and connect with other researchers. So you can, through seqr, you can connect to the Matchmaker Exchange. And then if you match with another researcher who's also submitted to the Matchmaker Exchange, you will see those details here. So for example, this researcher has submitted the BIC1 gene, which is associated with renal dysplasia. And I'd like to connect with this person. So you can use the contact host feature and can also track your conversations. So if I use the contact host, similarly, it auto-generates an email with relevant information about your case, and you can add anything else that you would like to convey and hit send. So this really you're able to do all of your analysis and leave notes, and which will be available to all members of your team so that you can really work collaboratively. All right. I also wanted to, hopefully your interest has been piqued and you really want to learn more about seqr. We have several YouTube videos that go over in detail and specific features, which we highly recommend. We also have an FAQ page, but this is really a good landing page for you to get started. I'll just open that up and show you what we have on here. So there's an FAQ page. We also have it available in Spanish, links to tutorial videos if you want to try out seqr. So essentially all you do is create an AnVIL account, and then you can just log into the main Broad Institute and try out the demo project yourself. Additionally, yeah, just more detailed information. There's another feature that I wanted to highlight up here is our feature updates. So it's a nice way to stay up to date when we launch new features. And we're quite excited about this. So late last year, what we launched was support with RNA-seq data. So really moving towards multi-omic data analysis within seqr. To get started with this, typically you would need to run publicly available pipelines. What we have here is Outrider and Fraser to generate your call sets. Those call sets would then be stored in an AnVIL workspace, and then you kick off the data loading process. And here's an example of what the plots would look like in seqr. So for example, if you have gene expression outliers, you can visualize that very quickly. So maybe if your case is negative from a variant perspective, you didn't find any variants of interest, you can just focus on expression outliers. So here, CSNK2B gene is a significant outlier. I can just use this link to find specific variants within this gene or any of these genes. Similarly, I can focus my search maybe on splice junction outliers, and here's another gene that has been flagged as an outlier. This has been incredibly useful to an analyst when reviewing cases. For example, you're reviewing all the variants, and side by side, you are able to see the particular gene expression. So, for example, you're interrogating a loss of function variant, you can actually see whether this gene expression has been reduced in seqr. Actually, I didn't mention when we did the variant search. We did a dominant search. I just wanted to highlight when it comes to gene expression. So yes, so here's the variant that we were looking at earlier. If you just click on the gene itself, it just pulls up additional information. So if you just wanted to do and links to other resources, databases, PubMed for literature search, gene disease associations, because there are lots of publications, but you really need to know how evaluate the strength of the evidence between the gene and the disease. So we have links to GenCC where they curate a lot of this information. This shared notes function can be really interesting because you can put a note here. I'm just gonna put this out. Yeah, I'm not gonna hit submit. But this note is going to be available to anyone using using seqr. So for example, maybe I'm interested in this gene and I'm collecting a cohort of cases. I can make a note of it over here, and which in a way informs other researchers who may be interested in this gene as well to maybe reach out to me so that we can collaborate. And at the bottom here, we have a gene expression data, which is from GTIX as well as isoform level expression. All right, I think I've touched on the main aspects of seqr. It's a really powerful tool for data sharing, collaboration, and doing your analysis. It's user friendly, and what that really helps is with a lot of researchers may not be computationally trained, so this really helps people who are more familiar with the phenotype themselves to participate in the analysis.